Hello everyone, this is Rohan Meshram and I am creating this demonstration video to demonstrate you practically as to how few few advanced software, or you can say how few advanced web server works for RNA structure prediction. Okay, so in this particular video we are going to see how two main popularly used web server works as such. Okay, as to how do you give them data and what is the kind of output that the server provides. Okay, so one of the most popular server used nowadays for secondary structure prediction of RNA molecules is in form of this RNA, or you can say M-fold algorithm. Okay, so this M-fold algorithm, you can find out the details of how this algorithm work. You can to know that you can refer this paper published in 2003, and as the title of this paper itself suggests, that it is a hybrid method that has incorporated multiple methods for predicting this RNA, or you can say secondary structure of RNA. Well, this being a server, well, it's its working is very simple. All you need to do is to provide certain sequence over here, which I have already downloaded from Mirbase. Okay, this is an miRNA sequence, okay, that I have downloaded from Mirbase, and it is from its nomenclature you can kind of figure out that this is a mice sequence of, say, some miRNA, and we are using this stem loop sequence. Okay, you can use uh, this identifier for Mirbase, and you can find out more details of as to what that miRNA sequence is. Okay, so all you need to give over here is the name of the sequence. You can give this miRNA. This is an miRNA sequence. Okay, and for time being we will keep all the values to default. Okay, so what it is doing, that this particular server assumes that we are going to fold your mRNA at a fixed temperature of 37 degrees centigrade. Okay, and it would be using certain parameters, okay, which are provided something over here. The temperature would be 37, and it would consider ionic condition of 1 molar NaCl with no divalent cation. As we have discussed in our previous theory lectures, that divalent cations are important for folding at tertiary level, and monovalent cations are important for creating this secondary structure. I hope you guys remember what has been taught in your first semester in okay, so we would keep all the other parameters for time being for at default condition. You can provide the email if you wish. It don't take much time, so uh, we will leave this box empty. Okay, and you can change the settings if you wish. Okay, so but for time being we would be using all the default setting and see how things work. Okay, so all you have to do is that paste that sequence, give your email ID if you wish, and hit this fold RNA button. Just wait for a while. It don't take much time. Okay, so the results are here. Okay, So now it says the output, it gives one of the output as energy dot plot. It outputs the dot plot. You can download it in various format: PostScript format, PDF format, PNG format, JPG format. Okay, maybe I will choose to see visualize my dot plot in PDF. Okay, so here is the dot plot. As you know that the sequence would fold upon itself, you are supposed to consider only either one of the triangle. Okay, so uh, this is it. Okay, this particular diagonal would correspond to the helix that is formed, secondary structure. This would indicate that there is a gap, and this would also indicate that there is a gap in your miRNA sequence. Okay, in your secondary structure that has been folded, right? So this is your dot plot representation of your secondary structure. Okay, and this is where you get the typical folded RNA molecule. Okay, first let us see that in this case it has provided a single structure, structure 1, but since it uses some hybrid approach, it is entirely possible that it might end up predicting multiple secondary structures which are equally probable. If that is the case, you would get here a list of those equally probable folded structures. Okay, and for each structure you are provided with the thermodynamic details as well. If you right click it and if you try to open link in the new tab, you would get all the thermodynamic details of this particular folded structure, or we will just come to this in a while. Okay, so if you remember that it is the similar thing that we have done in our theory classes. Okay, there is delta G values for external loop, whether there is stacking between G and C, whether there is stacking between U and G, and some free energy associated with that stacking. We have used the similar approach for obtaining the total free energy for, and we usually end up adding these things. Okay, so if you have forgot as to how these things are done, you are requested to refer the previous videos that I have already shared with you in your previous semester. Okay, so the free energy associated with this fold is minus 49.50. Okay, and we will come to this in a while. Okay, so next thing that we have is that you can download it the folded secondary structure in all these formats. There are a lot of formats that are available. So at least in Windows you can visualize the output in this PDF format, in PNG format, or JPG format. Let us open this secondary structure that is folded in new tab, or maybe new window, so that we can compare it with what has been given. Okay, so here is your result. You can see that some of the initial structure is that is not involved in any kind of helix formation or any kind of secondary structure formation. Okay, so this is the first residue in your RNA sequence, and helix or secondary structure formation in form of helix, or you can say the secondary structure formation start out from the second position. ठीक है, G at second position, base pairs C at 90th position, then subsequent C at first, second, third position might base pair with G at 89th position, and so on and so forth. You can see that there is a bulge over here. Okay, and then there is an internal loop over here. Okay, and so on and so forth. So this is a typical typical output of how these RNA sequence might fold. Okay, now let us try to compare. This is what I was talking about. Uh, Let us try and compare the thermodynamic details that you have obtained with the secondary structure that we have predicted so far. Okay, next. Okay, so here are the results for you know, your comparison. Okay, here it is. So initially we have got this external loop as shown over here. Okay, it is kind of not involved in any kind of secondary structure formation. Okay, so this is the free energy associated with it. Okay, then we have got stacking, as I have told. The G at second position, it forms base pairing with C at 19th position. So from over here you have got 1 2 3 4 and 5 staking, which correspond to 1 2 3 4. Okay, and the total free energy associated with this helix is minus 9.50. Okay, then you have got bulge over here. So if there is a bulge, then we might have some positive value. Okay, then again from here till here, it corresponds to the free energy associated, the values that are associated from the secondary structure that is predicted from over here to here. Okay, and therefore for this particular big helix, the free energy associated is minus 36.50. It contains 19 base pairs. Then you have got a big interior loop over here, and here is the free energy associated with it. Okay, and then you have got the big loop over here. Okay, sorry, a big helix over here. Okay, this is what the stacking is, stacking and the free energy delta G values that are associated with the base pairing that are involved in this stacking. Okay, and the total free energy for this particular helix is minus 15.3. Then you have got this bulge, which has got this delta G value associated with it. Okay, then again you have got two stackings which are shown over here, and finally you have got the hairpin loop, which is formed by these residues. Okay, so in this way you can associate how these delta G values might have been obtained for this secondary structure prediction. Okay, so you can download it for saving it if you want to visualize it further. Okay, uh, okay, so then what we have is the circular plots that we have discussed. You can download it in PNG format, in JPG format, or in PDF format. Okay, and make sure that you save all of your results in the given time, right? Because the results are usually erased after 12 hours of inactivity. So once you get your results, you are uh, advised to save whatever results you want in whatever formats. Okay, so this is how you use this mfold method. Okay, and you can refer that paper as to how parameters are to be what parameters are used for foldings, and what exactly is the algorithm behind this approach, or mfold approach as such. Okay, so next popular method that is or the next popular software that is implemented in RNA secondary structure prediction is this Vienna web server. Okay, so this RNA fold web server, or it is sometimes also called as the Vienna RNA web server for predicting the RNA sequence, RNA secondary structures as such. Okay, so it is also very simple. All you need to do is to copy this sequence, your input sequence, paste it over here. Okay, so now you can set a few algorithmic properties if you want, and you can also set your output option, whether you want interactive RNA secondary structure plot, if you want your results in form of mountain plot, or if you want to show the reliability annotation, so on and so forth. If you wish, you can provide your email. It is not necessary. Okay, so uh, if you provide your email after the job is completed, you would be notified on your email address, so that your job is finished, and you will be given with a link. If you click on that link, you would be taken to your result page as such. Okay, so the normal stuff that usually working with web servers as such. Okay, so we have pasted our sequence. We want the output in interactive RNA secondary structure plot, we want mountain plot and everything. Okay, all we need to do is to click proceed. It is just two-step process, and when the results are ready, you would be shown your results over here. Okay, here are the results. So over here the results are also shown in the very legendary dot and bracket notation. We have talked about this dot and bracket notation in our theory classes, and here is the dot and bracket representation of your RNA folding. Okay, so besides this, typical graphical output is also provided. Okay, and here are the thermodynamic properties. It is very similar to the ones that we have discussed in the Turner's method. Okay, so you can also download the dot plot as well. You can open it up in PDF. Okay, and here is the dot plot that has been generated. You have got your dot and bracket representation. Okay, uh, these people call it as centroid representation, this typical RNA secondary structures that you see. Okay, so this centroid plane structure drawing is what we are interested in. You can visualize that in a separate tab. Okay, a typical RNA secondary structure, as we have seen in case of this M fold as well. Where is it? Okay, I think we have opened it in some different window. Okay, this is dot plot. This is secondary structure prediction. You can also save the mountain plots, wherein the the plateau over here would indicate that there is loop, and the uh, and the and these you can see the details over here as to what exactly the mountain plot is. Okay, the height would indicate that this is a helix, and plateau would indicate that it is that this much region is involved in your loop. Okay, you can download this mountain plot as well in PDF format. Okay, so if you want to know more details of as to how this server works, what is the algorithm behind it, then you are referred to read this particular paper that was published in PNAS back in 2003 or 4, I suppose. Yes, it's published in 2004, and this web implication was published in 2008. So these are the two papers that you are advised to read if you want to go in more details of as to how free energy was calculated, and the other theoretical stuff if you are interested in. Okay, so you can, as you have opened it in new tab, you can save it so that you can visualize it later, or you can submit your results if you have been provided with an assignment. Okay, so this is how these two software works. Okay, so in theory we have discussed how your dot plot works, or how Nusinov-Jacobson algorithm works. Well, these two methods, mfold and Vienna websuit, well, these are quite quite complicated and extensive, so that would go out of the scope of our discussion. Okay, so anyway, if you want to explore more, I have already shown you as to where to look for. Okay, so if you want more details of this Vienna web server, then these are the two papers you are looking for. If you want more details of this mfold, then maybe this is the folder, or this is the paper that you would be looking for. Okay, so this is how you work on mfold and Vienna web server. Okay, and I hope it is quite very simple. You people might be already aware of how web server works. Okay, so this is it. Now I'm stopping my recording.